Pathophysiology of cardiovascular and pulmonary diseases. We are going to recite the preceding session with recitation of Holy Quran.
بسم الله الرحمن الرحيم والضحى والليل إذا سجى ما ودعك ربك وما قلى وللآخرة خير لك من الأولى ولسوف يعطيك ربك فترضى ألم يجدك يتيما فآوى ووجدك ضالا فهدى ووجدك عائلا فأغنى فأما اليتيم فلا تقهر وأما السائل فلا تنهر وأما بنعمة ربك فحدث.
Okay, kindly listen.
All of you hear this?
Speakers, can you hear?
Yes, clear.
भाई पहले एंटर करो, बहुत सारे लोग बाहर बैठे हैं। अच्छा चलिए।
Now rise for national anthem.
नहीं
Okay, now I would like to invite Professor Umar Ali Khan, who is President of Pakistan Physiological Society and Pro-Vice Chancellor of ISRA University, Islamabad, for the welcome address.
Assalamu alaikum wa rahmatullahi wa barakatuhu. So today we are very lucky to have Professor Ahmed Badr from Saudi Arabia. He will be presenting a plenary lecture on endothelial progenitor cells in vascular pathophysiology. And then we have got Dr. Ayaz Ahmed. He is my student and mashallah now he's professor. He will be talking about pathophysiology of arrhythmia. Professor Mehvesh Khalid, again another my student, a good, very good student also, that she did MPhil and FCPS both with me. And she'll be talking about advancements in precision medicine for cardiovascular disease management. Professor Sundus Tariq, who is also Vice President of SAP, he will be talking about Raynaud's phenomena, reviewing the pathophysiology. Dr. Kutsia Umaira Khan, I have a privilege to start her FCPS in physiology. And again, and a good student, heart failure, effectiveness of dietary supplements to boost mitochondrial metabolism in heart failure. Then our Yusra Hamid, Dr. Yusra Hamid, she is a trainee in physiology under the leadership of Professor Munazza, and she'll be talking about pathophysiology of myocardial infarction. Then postgraduate trainee of Professor Shazia from Islamic International Medical College, Dr. Aisha Mushtaq, will be talking about pathophysiology of asthma. So now I request Professor Ahmed Badr to please present his plenary talk. Professor Ahmed Badr, उन्हें rights दे दीजिए. Rights दे दी उन्हें.
Assalamu alaikum. Is my screen visible and my voice? Can you hear me? Hello. Someone has to answer, please.
Yes, sir, we can hear you. Assalamu alaikum wa rahmatullah.
No, this is not the group that is online.
Yes, we can hear you, Dr. Ahmed.
Dr. Dr. Omar, can you raise your hand, please?
Yes, we can hear.
Okay, wonderful. Thank you. Thank you so much. Thank you for inviting me, and thank you for giving me the honor to start the proceedings for today. And it is a great sight to see such a nice galaxy of physiologists. And I'm sure I can see from the participants that there are students as well. So I'll be keeping my short talk like we have been doing in the COVID days. That is half interactive and half, because of being online, we have some limitations. Anyway, so I will be addressing mainly the students, whether they are undergraduate students or they are the postgraduate students. So seniors, please do not mind if I ask some questions. So as is the norm that, I do not expect that I will be hearing your voices. So what you will be doing is this, that you will be keeping a piece of paper or your handheld devices, and you will be thinking of the answers to my few questions before I proceed. I have a bad habit that I stuck up on the title slide. I finish my lecture on the title slide, and then I proceed for the slides to have a revision. So, my dears, can you work out the title? What is meant by endothelial? What word is it coming from? Endothelium, probably. So just keep thinking in your mind. If I can hear any of you, this will be wonderful. But I'm not expecting that I am going to hear any of you. So I'll be going like that. So the first word is endothelial. And then what comes into our mind from progenitor? Especially the undergrad students. Have you heard this word, progenitor? Does any page of hematology and physiology comes into your mind? Progenitor, before.
Yes, sir.
Generation, something. Okay, so two words covered, endothelial and progenitor. And we come to the third word, that is cells. We all know about it. So this means there are some cells which are going to develop into what type of cell? Endothelial cells. So where are the endothelial cells located? I say I do not expect answer. Just answer yourself, okay? Just answer to yourself.
Blood, they are present in the walls of the blood vessels.
Wonderful. So they are present in the walls of the blood vessels. So endothelial progenitor cells will have something to do with what?
Differentiation of the endothelium.
All right, so we keep it very simple. That is, because they are the cells which produce endothelium. So in a fully developed human being, what will be their function? My question is not for the fetus. In a fully developed human being, what will be their function?
Basically, they can be used for any abnormality that has occurred in the endothelium and to repair that abnormality.
All right, so like I could sense from the answer, you are fully allowed in an online session to use your gadgets. Actually, it keeps us interested in the talk. Yes, so he has put it very simple in very simple way, that whenever there is some damage and there is need for some sort of repair, then these cells, they come into action. All right, so what is the commonest damage that you heard of or that you can think of in the blood vessels? or which blood vessels are subjected to?
Atherosclerosis is one of the.
All right, atherosclerosis is the mechanism, but what
Mhm.
will it lead to? What is the end point? All right, I know all of you can answer, so I'm not expecting just Dr. Ayaz, who is a very senior person, to answer. You know, I know that you are answering yourself, and your answer is actually ischemia, or your answer is hypoxia. So one of the types of the injury is ischemia or hypoxia. Can you think of some normal condition in the young boys who are nowadays getting less active due to these modern gadgets? Why I gave this sentence? Because I want to know, do you know the word neovascularization? Do you know of one physiological and one pathological condition in which there is neovascularization? So I know you can answer. All right, so I was referring to the physiological condition where we are trying to develop our muscles, and you know, we are proud of our muscles which have been built by different types of exercises in the gym and things like that. So new vessels, they are needed to support the developing muscles. Likewise, the pathological condition, we have got a tumor, God forbid, and this tumor is rapidly progressing, it is increasing in size, and it needs different type of nutrients, and for that it needs vascularization. So my dears, all these things, we have got these cells, and from here I will go a little sidetrack, and I will just try to recall with you what our teachers, you know, now the medicine has changed a lot, but Professor Omar Ali Khan, and we have got very senior persons here, like I can see Dr. Idris just now joining. He's my teacher. So, you know, in our times, the teachers, they used to tell this, that human being, like all the animals, we are made in a way that we can come over many diseases by ourselves, the inbuilt properties, what we call compensation in physiology. Although there are limits for compensation in physiology, but still, most of the animals, they do not get a chance to go to any hospital. They do not live with humans. They get injuries, they get different type of life-threatening diseases, and the fittest out of them, they survive. And the ones who are not fittest, they are not given treatments. They just die, or they are just put off. So this inbuilt mechanism is physiology, that is, which tries to prevent different type of threats to our body, or different challenges that we face. For example, I gave the example of exercise. My topic is endothelial progenitor cells, and we will go on slowly. The flow of presentation. Actually, I am doing a parallel job because I have been converted into host. I am entering people from the waiting room, and that is why I am pausing. There are many people in. I think there should be some co-host as well, because many people, they are waiting in the waiting room. All right. So we will define the endothelial progenitor cells. Then we will talk briefly about the discovery and the earlier studies, and we will talk about mechanism of action, how they perform their function. Because this is an ongoing research, then we will briefly touch current consensus. What are the things which are now established? Because this is a new thing. Then briefly touching quantification. Purpose is this, that those PG students who want to take up the projects rotating around EPCs, they could work out the places where there are methods for quantification available. We will touch the normal values, and I will suggest the avenues for research. Can you see my second slide, that is flow of presentation? Ayaz, you can answer, please.
Yes, sir. Yes,
Okay,
sir.
okay. So because the flow of slides, sometimes it gets blocked. All right. So I mentioned what is a progenitor cell. So all the undergrad students, they are familiar with this diagram, which is there in the very beginning of the hematology chapters. We are all aware of the word stem cells, and there was a peak or hike in the word stem cells, we started thinking that stem cells will be solution to every disease. And these EPCs are also, they are a product of that era, just before Obama became president in United States. And there was a hype in stem cells, and everyone was thinking that now every disease will finish and we will start using stem cells. So exactly in that period, the progenitor cells, they were found in the blood as well. So anyway, the stem cells, they go into symmetric cell division like hematopoietic stem cells, HSC. They can form their own form, that is symmetric cell division, or they can form another form that is called progenitor cell, which is actually parent cell. Stem cell is the grandparent cell. So progenitor cell is the parent cell. Yani even our endothelial progenitor cell, it will eventually originate from a hematopoietic stem cell, that is the grandparent cell. And then this progenitor cell, it can then divide into whatever type of tissue it belongs to, or whatever purpose it has been designated. So human endothelial progenitor cells, they are circulating cells that express a variety of cell surface markers similar to those expressed by vascular endothelial cells. I stop here for a little while, and please try to work out this word, the undergrad students specifically. That is, every cell has got some antigenic properties, and you have read it in your immunity. The body becomes the body reacts to the foreign cells based upon their antigenic properties, or the antigens which are present there on those cells, or the body goes against itself in autoimmunity. So this sort of thing. So you are well aware of the cell surface markers. Now in modern physiology, or in modern hematology, the cell surface markers, they are used to identify cells belonging to different areas of the body. So here as well, how we come to know that this particular cell belongs to the endothelium or the endothelial line, this is because they have got the surface markers which are similar to vascular endothelial cells. Now, the second thing is this: whenever there is hypoxia or ischemia, these cells, they go and they bind with the endothelium. And finally, they participate in the new vessel formation. So technically, the basic message of my presentation is up to here. Now, do you think that these cells, they will be present in circulation all the time? As I mentioned before, my target of the questions is basically the undergrad students, okay? So do you think that these cells must be present in the circulation all the time? How do we define circulation? From where circulation starts? So we know that bone marrow is not included in the circulation, but it is the place where our many cells, they are just sitting and waiting for the right time to come into circulation. So yes, these cells, they are not always present in the circulation. They come into the circulation whenever the body is faced with any type of challenge to the blood vessels, the biggest being hypoxia or ischemia, because the main function of the blood vessel is to supply with oxygen along with the other things. So my dears, going to the historical facts, just in 1997, not much time ago, the cells, they were discovered. And as I told you, that the basic thing based upon which they called them endothelial progenitor cell, it was the presence of specific markers CD34 and VEGFR2, that is the vascular endothelial growth factor. It was formerly called kinase insert domain receptor, or KDR. So I'm keeping it simple, not going into complexity so that I do not put off the listeners. That is because of the presence of surface markers, some cells, they were identified in 1997. Now, what is the significance of 1997? Now, the significance is this, that, people had free access to a new quantification or diagnostic method, or laboratory technique, that is flow cytometry. So in flow cytometry, people started experimenting with different things. When we were young, at that time, the new thing was, chromatography and electrophoresis. And at that time, people started experimenting with electrophoresis, and they found the types of thalassemia and types of hemoglobin and things like that. So now in this era of 1990s, they started experimenting on different fluids of the body and different tissues of the body and different cell cultures, and they started finding new things. Along with that, the cell culture, it became a highly developed thing, and you could actually produce new cells, and you could follow the cells, how they mature. Please try to understand this point. I told you stem cells are the grandparent cells, and then the progenitor cells, whatever type of cells they are the parent cells, and then there are the actual cells which are formed for a specific tissue. So now you could all develop in the laboratory these cell lines, what they call. That is, you could see the grandparent giving birth to different types of parents, which could make different types of tissues. And then you could see from these parents different type of offsprings that were specific and that were having features of their parents and less features of their grandparents. That is, here we come to the first mention of a population of peripheral blood. Now, please note this highlighted word, mononuclear cells. So they are mononuclear cells, more resembling monocytes. And as I will be picking up speed now to finish within my given time. So initially they thought that they are a series of the cells coming from the same series which give rise to monocyte and macrophages. So the early findings, they actually found in 2003 that these cells, they were able for producing new vascularization in experimental models. And the easiest way was what? I think my last question, so that I could pick up speed. So the easiest way was this, that they gave ischemia, artificial ischemia to the limbs of poor rodents, and they found that there is nevascularization, and they could pick up that in the tissues, the endothelial progenitor cells, they increased in number. So from here they proceeded, and they told that there are different markers, out of which CD34, CD133, CD45, and the vascular endothelial growth factor, they are present on these cells, and I'll be showing them to you in a minute. Then comes a landmark study in 2008, which exactly told that endothelial progenitor cells, they do not incorporate into the vascular endothelial monolayer by themselves, but they support angiogenesis. They do not directly participate in the formation of new vascular. So this brought us to the idea of paracrine function. You remember exocrine, endocrine, and paracrine? So they are producing some paracrine substances, which are then helping in the proliferative capacity of these cells. Now, then, as I told you, that you could see the different generations of the cells developing. So we came up with two names, early endothelial progenitor cells and late progenitor cells. And I will discuss this slide on this diagram, which is from the actual authors who published this in the Journal of American College of Cardiology. So you can see that these cells, they start developing from a common precursor, and gradually the markers, they start appearing. Here 133 and CD34. I go into early endothelial progenitor cells. Here another marker has come, that is CD14, and another marker has come, CD45, that is they are developing. Now CD45 and 14, they go on one side, and CD11 and 14, they go to another side. We call them early endothelial progenitor cells. They are very quick after an ischemic injury. 3 to 5 days, they come into circulation. From where? From bone marrow. They are heterogeneous, coming from different populations. They have got multiple markers, and they secrete different cytokines. But their in vivo angiogenic effects are low as compared with this late line, which comes into circulation in 7 to 14 days. They are more homogeneous. They have less markers, but at the same time, they secrete less angiogenic cytokines, because now they are in a in a position to start developing the endothelial cells, and they have got high angiogenic effect. Now, the same author, I'm sorry, because this is from Arabic side to the right, from the right to the left side because of my window problem. So he actually, exciting times, he said, we live in an exciting time. And this was 2008, because he thought, just like probably Dr. Aziz, he mentioned, he thought, and there were many studies now available who were thinking through the animal studies that now we can have therapeutic benefits of these cells. So quickly start thinking in your mind, what could be the therapeutic benefits? How could medicine benefit from the knowledge of these cells in depth? So now at that time, I'm still passing through the historical facts. So at that time, they thought that there are three basic places where you can find the EPCs. One, the hematopoietic. Second, the non-hematopoietic, which is in the umbilical cord blood or in the peripheral blood. In the beginning, they never thought that they are the same cells which are in the general circulation. And the important one, which I am mentioning here, they found these cells, sleeping cells, surprisingly, like macrophages, in umbilical cord tissue, in adipose tissue, in cardiac tissue, in neural tissue, and dental tissue. And to tell you truth, where not. They found these sleeping cells everywhere. They called it mature cells. So do not be worried about this slide. I will just pick up. The old concept was going something like this, that these cells from the stem cells, they pass through the monocyte route and they develop into macrophage-like cells which are in the tissues, fixed. And likewise, they actually found that there are mesenchymal stem cells which are converting into endothelial markers. And likewise, the tissue resident cells, like I told you in the cardiac cells, they can convert into endothelial markers. Yes, inshallah. I'm getting a call from Sobia. I think I have overshooted my time. So I'll be just skipping and I will be moving a little fast. Please do not give me another call. All right. So now the current consensus is this, that they are bone marrow derived cells which are used for endothelium regeneration, homeostasis, and neovascularization. And they proliferate in the bone marrow. They get mobilized from bone marrow. They come to rest in the endothelium where there is injury, and they lead to neovascularization. So as I have been told about the time, so I will be skipping a few things. The normal count in the normal healthy people, they are very low in the peripheral blood, but they increase as there is an ischemic injury. Aging is known to negatively affect the cell number. We have got many avenues where we can do the research. One is the general thing, that is, we can find correlations or association with different cardiovascular events and risk factors. Like, you know, nowadays, Dr. Tale and myself, we have two PhD students who are working on endothelial progenitor cells. One of them is working on different types of exercises and in volunteers of different types, that is, for example, obese and overweight and normal weight. And in different phases of menstrual cycle, she's working on the number or quantification of endothelial progenitor cells. Likewise, we have got a big thing in therapeutics where we can target the mobilization from the bone marrow and the recruitment sites. We can specify the recruitment sites by radio labeling. And likewise, we can develop different models of regeneration, repair, and neovascularization. So it is a very promising field for the researchers. And just for an example, I will tell you, like, for example, the researchers recently, they found in 2019 article, and this was the landmark article, that is the endothelial progenitor cells produced paracrine cells. They are in turn activating the neuronal progenitor cells to increase the proliferation of nerve fibers, which is expected because of the increased blood flow into the nerves. That's all. Thank you very much. Sorry for taking a little longer than my time. Thank you.
Thank you so much, sir, for such a knowledgeable, informative presentation. Now, the next presentation is from the Professor Dr. Ayaz Ahmed from Muhyiddin Medical College. Title is Pathophysiology of Arrhythmia. Sir, you have 10 minutes for this presentation.
Dr. Ayaz, can you hear? Yes, Professor Ayaz, please start.
Assalamu alaikum, sir. Assalamu alaikum, sir. Yes, sir. I'm just going to start. There are some internet issues. And basically, I am right now, I am in Azad Kashmir. So, Assalamu alaikum wa rahmatullah. I hope that all of you can hear me.
Yes, please.
And my presentation is visible.
Yes, please.
Yes.
Okay, sir. First of all, I am thankful to Professor Umar Ali Khan and Pakistan Physiological Society to provide this opportunity to me to present this topic to you. Basically, this topic is cardiac arrhythmias, which I am going to present today. And as I have told that my name is Dr. Ayaz Ahmed, and I am working in Muhyiddin Islamic Medical College, Mirpur, Azad, Kashmir. First of all, these are some of the objectives which I will try to achieve during this presentation. I will describe the normal physiology of the cardiac rhythm, then we'll define the cardiac arrhythmias, then some of the pathophysiology, the common pathophysiology of the development of the cardiac arrhythmias, and the relationship of these cardiac arrhythmias with the ECG. So first of all, what is normal rhythm? We all know that basically the heart rate, if it is between 60 to 100 beats per minute, and if the every impulse of the heart is generated from the SA node, and if it normally propagates through the normal conducting pathway of the conducting system, and it has the velocity according to the each part of the conducting pathway, then it is called the normal rhythm. Just to elaborate this thing, that basically this is the SA node, and the normal impulse should generate from the SA node. It should propagate through both the atria, that is the right atrium and the left atrium, and then it should pass through the AV node, which is the electrical window between the atria and the ventricle, which is the only electrical window. So it should pass through the AV node, and after passing through the AV node, as it passes through the left bundle branch, it should depolarize the septum, and after that it should depolarize the apex of the heart through the left bundle branch, and then through the right bundle branch, and then the base of the heart. And the speed of this conduction should be according to each part. That is, it should be slower in the AV node, and it should be fast in the Purkinje system. So this is about the normal impulse generation, and it will result in the normal cardiac rhythm. Now, what is this is the ECG of the normal sinus rhythm. We are aware of this, and we can see that the configuration of this ECG, that there are the P waves, there is the QRS complex, and then there are the T waves, and then there are basically different isoelectric lines. And its rhythm should be normal, it should be regular, and then there are certain time periods of these different complexes and the waves, and it should also be within the normal limits. Now, what are the cardiac arrhythmias? Any deviation from these things, these principles, it results in the cardiac arrhythmia. That is, if the heart rate it's less than 60, or if it is more than 600 beats per minute, or if it is not being generated from the SA node or any other part of the heart, it is being generated from any other part of the heart, and or its velocity is not normal, so and it is not following the normal conducting pathway, then it will result in the generation of the cardiac arrhythmia. There are basically three basic mechanisms which result in the cardiac arrhythmia. One is the alteration in the normal automaticity, then is the triggered automaticity, and the other one is the re-entry or the circus movement. These are the three basic mechanisms for all the cardiac arrhythmia. So I will explain one by one for what is the alteration in the normal automaticity. We know that the pacemaker cell of the heart is the sinoatrial node, and basically this node has some special characteristics which are as far as action potentials are concerned. Number one is that it has an automaticity, means that it can generate its action potential without any external influence, and this is due to these channels, which are the sodium leak channels, which continually leak into the cells of the assay node, and these basically results in the generation of this slope, which brings it toward the threshold potential, and then there is the entry of the calcium ions, that is the voltage gated calcium ion, and then repolarization is basically due to the potassium ion. So anything, if it disturbs this automaticity, it can result in the cardiac arrhythmia. For example, if we talk about the normal things like the sympathetic stimulation, due to any reason, this sympathetic stimulation will fasten the entry of these sodium leak channels, and the time of this slope will be decreased. So as a result of this decreased in the slope of the time, the number of the action potentials generated in the assay node in that time period will increase. And opposite to this, if there is a vagal stimulation, the vagus nerve, basically it results in the hyperpolarization. That is, these repolarization phase then is prolonged, and it results in the hyperpolarization, that is more exit of the potassium ions. So it is difficult for the cells to come back to the threshold level, and takes more time, and that results in decreased in the number of impulses in that time period. So this is the phenomena of the alteration in of autorhythmicity. Then the trigger automaticity means that the any other cell then the assay node takes over the automaticity. For example, that cell can be the atrial cell, or it can be a ventricular cell, or it can be a AV nodal cell, or it can be AV node as well. So due to some abnormal membrane properties, that is due to ischemia, injury, or due to loading of these atrial cells or the ventricle cells with the cation, if excessive cations are loaded due to catecholamines, these cells results in they take over the automaticity, and they can result in the cardiac arrhythmias. So again, this triggered automaticity, this is the basically action potential that is present in the ventricular muscle cell or the atrial muscle cell, and we are aware of the different phases of this action potential. That is, depolarization is due to the sodium ion, then there is a plateau formation, that is due to the calcium ion entry and the potassium ion exit, that basically results in delayed repolarization, and then at the end calcium ion entry ceases, and then potassium ion exit continues, and it results in repolarization. So any abnormality, for example injury or trauma or anything to this cell, basically can result in automaticity. And there are the two types of this automaticity. One is early after depolarization, that starts earlier before this repolarization ends, this repolarization end, and then is the delayed after depolarization, that can occur when the depolarization has ended and the new impulse, which is not a normal site of the impulse generation, starts generating the impulses. So this is the trigger automaticity. The third phenomena is the re-entry or the circus movement, and basically again it is due to some scar tissues that are present in the atria or the ventricle. This is the normal pathway for any impulse. This is basically a cross section of the cardiac tissue, and we can see that these cells basically they start the action potential from here, and normally these action potentials go over there, and these action potentials when reach over this area is refractory. That is absolutely refractory period, that it cannot generate the new action potentials. So this impulse cannot travel again through this pathway. But what happens in the scar tissue, that this impulse travels again and again through this area, and it results in the circus movement, and it results in the generation of the cardiac arrhythmias. So the recircuit entry, it can be due to long pathways that occur usually in the dilated heart. The long pathway, when the impulse comes back to this area, that area is no longer refractory to that impulse, and the new impulse generates over there, and it continues. Or it can be due to low velocity of the conduction, that is slow velocity of the conduction, and when that impulse reaches at that point, that impulse is no refractory at that period, that can and can generate into the new impulses. That is, it can be due to ischemia or the conduction blockade, or due to increased extracellular fluid potassium concentration. Or there are the shortened refractory period of the muscle, that can be due to epinephrine or repetitive electrical stimulation of that part of the muscle, that results in the shortened refractory period and the generation of the new impulse, resulting in the arrhythmias. So this is a very simple classification of the different arrhythmias of the heart. First of all, what we have said that about the normal, that the heart rate should be between 60 to 100. Its origin should be from the SA node, and it should follow the normal conducting pathway and normal velocity. So anything between about the heart rate between 60 to 100 is normal. If the heart rate increases from 100 to 150, then it results in the simple tachyarrhythmias. If heart rate is between 150 and 250 beats per minute, then it results in paroxysmal tachyarrhythmias. It is called paroxysmal because it suddenly onsets and suddenly disappears. Then if the heart rate is between 250 to 350, then it results in the flutter. Again, it can be atrial as well as the ventricle in origin. Then if the heart rate is more than 50, it can result in the fibrillation, and again, it can be atrial or the ventricle. So this is about the increase in the heart rate. If the heart rate is less than 60, that is between 40 to 60, then it is called the mild bradyarrhythmias. And if it is between 20 to 40, it is called the moderate bradyarrhythmias. And if it is less than 20, it is called the severe bradyarrhythmias. So this was the one way to classify the arrhythmias. The other way of the classification of the arrhythmia is the site at which these arrhythmias are generated. For example, if these arrhythmias are being generated from the SA node, these are called the sinus arrhythmias. If these are being generated from the atrial node, atrial tissue, this is called the atrial arrhythmia. If these are being generated from the AV node, these are called the junctional arrhythmias. And if these are basically generated from the ventricular tissue, these are called the ventricular arrhythmias. And the pathology will remain the same. That is due to any ischemia, injury, or pathology, or any other thing that can result, or some drugs or some chemicals that can result in alteration in the automaticity, or basically triggering the automaticity that results in the generation of these arrhythmias. So sinus arrhythmia, we are well aware of this sinus arrhythmia. That is, it is the fluctuation in the heart rate with the phases of the respiration. That is, during inspiration, the heart rate increases because during inspiration, the vagus nerve is inhibited. And once the vagus nerve is inhibited, the heart is out of the inhibition. And that results in increase in the heart rate. And during expiration, the vagus nerve is stimulated. And once vagus nerve is stimulated, it basically depresses the SA node and the AV node, and heart rate decreases. So it is a normal phenomena that alteration occur. This alteration normally can occur in the normal respiration, and it is called the heart rate variability. The important point, the clinically important point in this is that some people who have diabetes mellitus, they will not have this heart rate variability. So due to autonomic neuropathy, this fluctuation will not be present. And similarly, the persons who have transplanted heart, they do not have the normal vagal innervation. So their heart will also not show this heart rate variability. Then these are the ECG changes that occur in the sinus arrhythmia. We can see that during inspiration, these complexes, ECG complexes at the given period of the time are more as compared to the expiration, where these complexes are, the number of these complexes are less. Then the sinus tachycardia, we can say that when the heart rate is more than 100, and that can occur during the exercise, during the emotions, fever, or during hyperthyroidism. And this is a comparison of a normal ECG and a ECG of sinus tachycardia. We can see that at a given length, the number of the complexes in the normal ECG are 8, while in the sinus tachycardia, these complexes are 13. So rest of the configuration of the ECG waves, that is P, Q, R as complex and T waves, are totally normal.
Dr. Ayaz, your time is over. Sorry
बस
to
मैं 2 मिनट
interrupt you. Okay, thank you.
Sinus bradycardia, again, we can see that in this ECG, again, it can be due to sleep, athletes, or normally it can occur in athletes or due to some drugs. And again, we can see that the number of the complexes this is the normal ECG, where the number of the complexes are 8, while in this in the below the ECG, which is here, basically shows the number of complexes are only 5. Then the tachyarrhythmias, as I said, that basically the tachyarrhythmias can be paroxysmal tachyarrhythmias, these can be flutter, this can be fibrillation, this can be from the atria, and these can be from the ventricles. If these tachyarrhythmias are from the atria or the AV node, then these are called the supraventricular tachycardias, and if it is denoting from the ventricles, these are called the ventricular tachycardias. The mechanism remains the same, that is the re-entry circuits or any irritable multiple foci that can result in the generation of these tachyarrhythmias. So this is basically a supraventricular tachycardia, then as the atrial flutter and the atrial fibrillation. The problem with the atrial fibrillation and atrial flutter is that the number of the P waves are more as compared to the QRS complexes. And some of these P waves or some of these conductions from the atria, they pass through the AV node and they result in the impulse generation in the ventricles, resulting in the formation of the PVCs, that is the premature ventricular contractions. Again, these are the things about the ventricular tachycardia, ventricular tachyarrhythmias, that is the ventricular tachycardia, ventricular flutter, and ventricular fibrillation. And basically, there is no association between the ventricle and the atria, and it results in decrease in the cardiac pumping because the heart cannot fill with the blood properly. So again, if I summarize my presentation, basically there are many reasons of the tachyarrhythmia, but the basic mechanism is that there is alteration in the normal automaticity of the heart. There is a trigger automaticity, that is the pacemaker cells. They do not control the conducting system. Instead, some other tissue of the heart takes over the control, or there are some re-entry circuits. And this is the classification which I have basically explained. So this was about my presentation. Thank you very much.
Okay, thank you so much, Dr. Ayaz. Such a wonderful presentation. Now, the next presentation is by Professor Dr. Mavish Khalid from Akhtar Said Medical College. Topic is advancement in precision medicine for cardiovascular disease and management. Dr. Mavish, you have 10 minutes for your presentation. उनकी आवाज नहीं आती
Bismillahirrahmanirrahim. Assalamu alaikum. Can you all hear me?
जी आवाज आ रही है
Okay, ji. I'm Dr. Mavish Khan from the Department of Physiology, Akhtar Said Medical College, Rawalpindi. Today, I'm going to quickly highlight the latest developments in the personalized medicine approaches that are aimed to tailor the diagnosis, treatment, and prevention strategies for individual patients based on their genetic makeup, lifestyle, and other relevant data. Cardiovascular disease remains the leading cause of death worldwide, accounting for 32% of all global deaths, with the expectation that this number will rise to more than 23.6 million deaths annually by 2030. Precision medicine is basically an integrative approach to cardiovascular disease prevention and treatment that considers an individual's genetics, lifestyle, and exposures as determinants of their cardiovascular health and disease phenotypes. Precision medicine incorporates standard clinical and health record data with the advanced panomics. This advanced panomics basically includes transcriptomics, which is the study of RNA, its structure, its function, its regulation, its expression. Epigenomics, which is basically the study of epigenetic changes in the cells, the changes that occur when a gene is switched on or switched off without changing the original DNA sequence. Proteomics, which is the study of proteins, their structures, their functions, their interactions. Metabolomics, which is basically the study of metabolites in our body, the metabolites that are present in our cells, body fluids, etc. This advanced panomics also includes the study of microbes that are present in our body. They are present and they are beneficial to us. So this advanced panomics is basically including this transcriptomics, epigenomics, proteomics, metabolomics, and microbiomics for phenotyping. Regarding the development of various personalized medicine approaches, it is important to mention the role of genomic medicine, because genetic variations can contribute to the development, progression, and response to treatments in cardiovascular diseases. Emerging genetic testing techniques and next-generation sequencing technologies enable efficient and cost-effective analysis of various genetic variants, like those associated with conditions like coronary artery disease, hypertension, and familial hypercholesterolemia. Biomarkers also play a very important role in determining the personalized medicine approaches for cardiovascular disease management. These biomarkers, which are basically the measurable indicators in the body that provide information about an individual's disease risk, progression, or treatment response. Biomarkers such as high sensitivity troponin, C-reactive protein, and natriuretic peptides, they have a role in identifying individuals at high risk for disease risk assessment. The predictive value of biomarkers can provide insight into disease progression and guide treatment decisions. Combining genetic information with biomarker analysis enhances risk stratification and personalized treatment planning. Nowadays, the advanced non-invasive imaging techniques such as cardiac MRI, CT angiography, and 3D echocardiography provide detailed anatomical and functional information to identify structural abnormalities, vulnerable plaques, and atherosclerosis progression, enabling targeted interventions and precise diagnosis of cardiovascular conditions. Regarding a few precision therapies and targeted interventions, I would like to mention here the role of individualized drug selection depending upon the genetic profiles and biomarker analysis. By understanding an individual's genetic profile, healthcare providers can make more informed decisions about drug selection, dosage adjustments, and potential drug interactions, leading to personalized medicine. Biomarker analysis involves assessing specific molecules or indicators in the body, and that can provide information about a person's disease state, drug response, or treatment outcome. This information can guide healthcare professionals in selecting the most appropriate medication for an individual, improving treatment outcomes and minimizing the risk of adverse reactions. There is an important role of pharmacogenomics in optimizing drug efficacy and minimizing adverse effects of the drugs. Pharmacogenomics is basically the study of how an individual's genetic makeup influences their response to medications. It involves analyzing the interaction between genes and drugs to understand why different individuals respond differently to the same medication. By identifying genetic variations that impact drug metabolism, drug targets, or drug interactions, pharmacogenomics helps to optimize drug therapy and minimize adverse effects. By integrating the pharmacogenomics into clinical practice, healthcare providers can enhance treatment outcomes and improve patient safety. It allows for a more precise and personalized approach to drug therapy, ensuring individuals receive the medications that are most likely to work for them while minimizing the risk of adverse reactions. The novel therapies such as DNA editing and RNA-based therapeutics, they are also nowadays playing a very important role in personalized medicine approaches in cardiovascular disease management. Gene editing, which involves modifying an individual's genetic material to correct or modify disease-causing genetic mutations. By precisely editing these genes, it may be possible to address underlying genetic causes of cardiovascular diseases, potentially leading to more effective treatments. RNA-based therapeutics utilize different types of RNA molecules to modulate gene expression and protein production within cells. These therapies can be designed to target specific genes or proteins involved in cardiovascular diseases. For instance, small interfering RNA can be used to inhibit the production of certain proteins associated with heart diseases, while messenger RNA can be used to deliver instructions to cells for producing therapeutic proteins or antigens. Both gene editing and RNA-based therapeutics present promising avenues for the development of novel treatments in cardiovascular disease. These approaches are still being researched and refined. They hold the potential to revolutionize the management and treatment of cardiovascular conditions by directly targeting the underlying genetic factors contributing to these diseases. We cannot put aside the role of lifestyle modifications in precision prevention. Precision medicine plays a crucial role in designing personalized lifestyle modifications, including exercise, diet, and stress management to reduce the risk of cardiovascular disease. For example, genetic information may reveal certain genetic variations that influence how a person responds to exercise or metabolizes certain nutrients. Based on this knowledge, healthcare providers can design exercise programs that are better suited to an individual's genetic profile, taking into account factors such as optimal exercise intensity, duration, and type of physical activity. Understanding genetic variations related to nutrient metabolism can help tailor dietary recommendations, ensuring that individuals receive personalized nutrition guidance that maximizes cardiovascular health. Stress management is a very important aspect of reducing cardiovascular risk. Genetic and biomarker information can help identify individuals who may be more susceptible to stress-related cardiovascular problems. By incorporating this information, healthcare providers can develop personalized stress management strategies, such as mindfulness techniques, relaxation exercises, counseling that are more suitable for the individual's specific needs. An important consideration and challenge regarding implementation of personalized medicine approaches is taking care of ethical, legal, and social implications of these personalized approaches. Personalized medicine raises ethical questions surrounding privacy, consent, and potential discrimination. Collecting and analyze genetic data can reveal sensitive information about an individual's health risk, predispositions, and even their family members. Ensuring the ethical use of this information involves obtaining informed consent, protecting privacy through secure data storage and transmission, and establishing policies that prevent misuse or discrimination based on genetic information. Robust data protection measures, secure storage systems, and strict data sharing protocols are necessary to safeguard privacy and prevent unauthorized access to or misuse of genetic data. One of the challenges is ensuring equitable access to genetic testing. Genetic testing can be expensive, and not all individuals have equal access to these tests due to financial constraints, limited availability in certain regions, or lack of awareness. Addressing this challenge requires efforts to improve affordability, expand access to genetic testing services, and provide education and awareness about the benefits and limitations of genetic testing to empower individuals to make informed decisions about their health care. Ensuring equitable access to precision medicine approaches is crucial to avoid existing
Dr.
healthcare
Mavish?
disparities.
Dr. Mavish, sorry
Just
to interrupt
one
you.
more slide.
Okay, thank you.
It is essential that all populations, regardless of socioeconomic status or geographic location, have equal opportunities to benefit from precision medicine. Ongoing research in precision medicine for cardiovascular diseases, continuously exploring innovative technologies and approaches. One significant area of advancement is the utilization of artificial intelligence and machine learning. AI and machine learning can analyze large data sets, including genetic information, biomarker profiles, medical imaging data, and electronic health records to identify patterns, correlations, and predictive models that help in risk assessment, disease diagnosis, treatment optimization, and patient outcome predictions. It is important to continue research and validation to ensure the reliability and clinical applicability of these advanced technologies. Precision medicine has the potential to improve patient outcomes while reducing healthcare costs by guiding personalized treatment decisions, implementing targeted prevention strategies, avoiding ineffective treatments, enabling early detection and intervention, and optimizing resource allocation. Incorporating genetic predispositions and biomarker profiles into cardiovascular prevention strategies allows for more targeted and personalized approaches to disease prevention. Ongoing research, integration of technologies, and broader access to precision medicine approaches are essential to realize its full impact on healthcare delivery. Thank you so much.
Okay, thank you so much, Dr. Mavish. You have enlightened every aspect of your topic. Now, the next topic is Raynaud phenomena, reviewing the pathophysiology by Professor Dr. Sundar Tariq from the University of Islamabad, who is also the Vice President of SAP.
Thank you so much for inviting me. I'm really thankful to all the organizers, especially the President of Pakistan Physiological Society, Professor Dr. Umar, for giving me the opportunity to present here. Am I audible to all?
Yes.
Yes, madam.
Okay. Thank you for the invitation. I will be presenting today on Raynaud's phenomena, reviewing the pathophysiology. And my objective will be to define the Raynaud's phenomena and to explain the pathophysiology in relation to various management options that we can adopt. So, well, I like to disclose that I do not have any conflict of interest. Going to the definition of Raynaud's phenomena, We are all are very well aware that this is a multifactorial vasospastic disorder that is characterized by a transient, recurrent, and reversible constriction of the peripheral blood vessels. This phenomena was initially discovered by a French physicist, Maurice Raynaud, and he presented this as a part of his doctorate thesis, local asphyxia and symmetrical gangrene of the extremities. And he initially defined it as an episodic, symmetric, acral vasospasm characterized by pallor, cyanosis, suffusion, and a sense of fullness or tautness, which may be painful. So taking his idea forward over a period of 150 years, a lot of scientists have contributed in understanding the pathophysiology of this Raynaud's disease. There are two terms that are used synonymously for Raynaud's syndrome or Raynaud's disease. We will see what is the difference between the two. So the term is actually Raynaud's syndrome, and this is further like divided into two: Raynaud's disease and Raynaud's phenomena. So Raynaud's disease is a relatively milder form of the disease, and this is also called as the primary Raynaud's syndrome or primary Raynaud's phenomena, while the Raynaud's phenomena is also termed as the secondary Raynaud's syndrome. So Raynaud's disease is a milder form. Its pathophysiology is usually idiopathic, and this is more common as compared to the Raynaud's phenomena. While the Raynaud's phenomena is also termed as a secondary Raynaud's syndrome, and this could lead to a more severe form, and this is usually linked to the underlying disease conditions, medications, or various lifestyle factors, and this is less prevalent in our society. So this disease or this phenomenon is characterized by a triphasic discoloration. Initially, it presents with white digits. White digits, which is also called as ischemic digits, is there because of the intense vasoconstriction. This is followed by the second phase, which is called the cyanotic phase, in which there are blue digits, and this is because of hypoxemic venous stasis and presence of the deoxygenated blood. And then the third phase is the hyperemic stage, hyperemic phase, or the red digits, which is because of hyperemic reperfusion. So the first two phases stays here for almost like 20 minutes, followed by the third phase. And in certain patients, we may not found the second and the third phases, and we can only make the diagnosis based on the white digits or the ischemic phase of the Raynaud's phenomenon. So going to the pathophysiology, we can see that this phenomena is linked to two basic pathophysiological mechanisms. One is the exaggerated vascular response, and other is the stimulation of the sympathetic nervous system. So the pathophysiology involves two basic phenomena. One is the vascular response, and the other is the nervous response. And these are initiated in response to the various factors. The most important one of them are, number one, the relative shift from warmer to the cooler temperature. So cold temperature is a very common factor that can contribute to the initiation of the Raynaud's phenomena. And then the second one is stress, both physical as well as the emotional stress. And then we can have some idiopathic local defects to the extremities like the systemic sclerosis or scleroderma that can also lead to initiation of the vascular as well as the nervous responses. So in response to the vascular as well as the initiation of your sympathetic system in response to the stress, we can see that there is actually vasospasm in the extremities. And these extremities can involve the fingers, toes, ears, nose, face, knees, and the nipples. So as response to the vasoconstriction, obviously there is going to be decreased blood flow into the extremities, and this can lead to initially sensation of numbness or pins and needles. And this ischemia will lead to appearance of the pallor on the extremities, and that's why they're called as the white extremities. Then in response to this, there is going to be stagnant hypoxia. The oxygen that is extracted from the blood sitting in the vessels, it is actually deoxygenated blood, and this leads to cyanosis or blue extremities. Then the third response is going to be, as we are very, very well aware, that whenever the local blood flow to the tissues is compromised, two mechanisms are going to be activated. the metabolic theory and the oxygen demand theory. So there is production of the anaerobic metabolites, and these anaerobic metabolites, we know there are a lot of anaerobic metabolites, lactate, adenosine, ADP, etc. And secondly, there is going to be decreased oxygen and nutrients to the arterioles. Both of these are going to lead to cessation of smooth muscles vasoconstriction. So now, because of this response, the blood vessels are going to be dilated and blood re-enters into the extremities, leading to the third phase, which is a reactive or hyperemic phase in which the extremities becomes red. So these are the three basic phenomena of the Raynaud's disease. If these are the milder form, if the disease becomes more severe and if the disease stays there for longer periods of time, what happens is there is chronic decrease of oxygen and nutrients to the digits. This can lead to finger pulp atrophy, and this chronic deficiency of nutrients will lead to chronic decreased perfusion to the digits, and this can obviously lead to tissue ischemia and infarction, further leading to digital ulcerations and gangrene. Then we can also see the appearance of mottled purplish discoloration of the skin because of the swelling of the venules and stagnation of the blood in the peripheral vessels. Going into the depths of the pathophysiology, why this vasoconstriction occurs and why there is stimulation of the sympathetic nervous system, we will briefly see it. So we can see that there are certain factors like we have prostacyclins and prostaglandins in the body. The prostacyclins are responsible for causing vasodilation. And whenever the levels of these prostacyclins decreases, this is going to cause vasoconstriction. So in the patients of Raynaud's disease, it has been seen that the levels of prostacyclins are less. That's why if we use prostacyclin analogs as a treatment option, we can see that the patient benefits from these drugs. Secondly, we have another important factor, endothelin-1. So endothelin-1 is usually, we know that this is a potent vasoconstrictor and is a profibrotic agent, and its levels have been found to be increased in patients of, again, Raynaud's phenomena. And if we use endothelin antagonist, this is going to again benefit the patient. Then we have other vasoconstrictors like angiotensin II. Its levels have also been found to be more in patients of Raynaud's disease, causing vasoconstriction. Then it has also been observed by various scientists that the calcium mobilization in the smooth muscle cells also increases in patients of Raynaud's disease, causing the smooth muscles to become more active, and there is more vasoconstriction. So these patients will also benefit from calcium channel blockers that can lead to vasodilation and then will benefit the patient. Then we have another important factor, calcitonin gene-related peptide. Physiologically, this agent is responsible for causing vasodilation. This actually increases the levels of adenylate cyclase, increasing cyclic EMP, and is responsible for relaxing the smooth muscles of the blood vessels. So if its levels decreases, which actually happens in Raynaud's phenomena, this is again then going to affect the vasodilation. There will be intense vasoconstriction, and again, the three phases of Raynaud's disease. Then we have other factors like nitric oxide. Its levels have also been found to decrease in Raynaud's disease. And then scientists have also found that there are some other intravascular factors that can play the part in the pathophysiology of the Raynaud's disease. And in them, the most important one is platelet activation. So the platelets are also activated. This has been seen that there are increased levels of thromboxane A2, there's increased levels of thromboxane synthase, and all of them are helping in platelet aggregation. So this is going to again cause increased platelet aggregation and decrease in the flow of the blood, sluggishness of the flow, increased blood viscosity. All these factors are again going to contribute towards ischemia, towards the tissues. And then we've also seen that there is generation of reactive oxygen species that also damages the endothelium as well as the red blood cells, and that also plays a part in platelet activation. So all these factors are contributing as an intravascular additional factors that can worsen the Raynaud's phenomena. Then the last most important factor is nervous regulation, nervous activation. So what happens that whenever we have extremes of stress, emotional or physical, there is going to be activation of the sympathetic nervous system. So it has been seen that the two adrenoceptors, the alpha-1, especially the alpha-2C type of adrenoceptors, are increased in patients of Raynaud's phenomena. And whenever we have increased translocation of these alpha-2C type of adrenoceptors on the membranes of the blood vessels, it is going to cause increased response to norepinephrine and then again vasoconstriction. So if we use some blockers that can block these receptors, and again, we can benefit from this treatment option. So based over a period of research of 150 years, scientists have come up with a lot of pathophysiology, and now they have used that pathophysiology to decrease the prevalence of this disease and to increase the treatment options that are available for this disease. The most important one still remains that we advise the patient that they should avoid exposure to cold, and there should be proper emotional regulation so that the disease cannot be worsened or its incidence can be decreased. Then we have further treatment options that I have discussed subsequently. So this is all about my presentation. Thank you so much. If there are any questions, I'm here to answer.
The question answer. Thank you so much, Professor Dr. Sundas, for such a wonderful presentation. Now the next presentation is by Associate Professor Dr. Qudsiya Umaira Khan from CMH Lahore Medical College and Institute of Dentistry. Topic is heart failure, effectiveness of dietary supplement to boost mitochondrial metabolism in heart failure.
Assalamualaikum. First of all, I would like to thank you, Professor Dr. Omar Ali Khan, Dr. Sobia, for inviting me to present in this prestigious seminar, and I am truly honored. So today's topic is heart failure. This is one of the earliest terminologies which I listened when I was a child, heart fail. My mom always used to say कि आपसे कोई गलती हो गई तो heart fail हो जाएगा. So when Sobia asked me to present in this seminar, my heart was almost failing, but I just gripped up myself and I thought कि कुछ लोगों से मैंने पूछा, उन्होंने कहा कि कोई सुन नहीं रहा होता. तो I just thought कि कैसे इस topic को हम ये बनाएं कि everybody should be listening to this topic. So let me
a very good
start off with this topic.
A very good start. No, you made all of us to listen carefully.
Yes, yes, I just want all of you to listen. कि कोई सुन नहीं रहा था, जो मर्जी बोल देना. So anyways, heart, I have just gripped up my heart and I was very much conscious that Professor Umar is listening. So Bismillahirrahmanirrahim. Any heart condition that reduces the ability of the heart to pump enough blood to meet the body's need. It can be caused by damaged heart valves, external pressure around the heart, vitamin B deficiency, primary cardiac diseases, or any other abnormality that makes the heart a hypoeffective pump. There are certain definitions. I've gathered these definitions from the net and other books, that it is the inability of the heart to pump blood at a sufficient rate to meet the metabolic demands of the body. For example, oxygen, the cell nutrients at rest, and during efforts to do so only if the cardiac filling pressures are abnormally high. It's a complex clinical syndrome characterized by abnormalities in cardiac functions and neurohumoral regulations, which are accompanied by effort, intolerance, fluid retention, and a reduced longevity. So there are other definitions, but it's generally heart failure is a clinical syndrome, and that is a result of certain underlying diseases which could be related to the heart itself, or it could be related to other structural diseases. If we come to the stages of the heart failure, there are certain patients that are at high risk of heart failure. Then we have patients that are number 2 category, they are the pre-heart failure stages or stage B. Then we have the symptomatic heart failure, that is the stage C. And then we have the advanced heart failure, that is the stage D. There are certain causes or etiologies that are related. A person having a heart attack, which could be due to angina, myocardial infarction, that can result in a cardiomyopathy. Then we have got valvular cardiomyopathies, hypertensive diseases, inflammatory cardiomyopathies, metabolic, general systemic diseases, muscular dystrophies, neuromuscular disorders, and then sensitivity and toxic reactions, peripheral cardiomyopathies. So a heart failure that results from hypertrophy or restricted diseases that can be related to the right side of the heart, could be related to the left side of the heart. Heart failure can be systolic, diastolic. It can be compensated or decompensated, a high output failure, a low output failure. It could be acute condition. It could be a chronic condition. So this is another slide which is showing you the diastolic or the systolic features. I hope this bar is not interrupting in between. Then, sorry. Then this is a chart I found it very effective in explaining the definitions, the stages and classifications of the chart. And you can see that if we defined heart failure, it's a clinical syndrome with current or prior underlying conditions. It could be related to the different stages, and then we can classify the heart failure according to its ejection fraction. If the ejection fraction is reduced to less than 40%, or if it is between 41 to 49%, or if it is a less than 50%. So what are the physiological consequences of having a heart failure? Serious physiological consequences of heart failure could be, there could be increased workload on the damaged heart. There could be overstretching of the heart, which further weakens the heart. There could be filtration of fluids into the lung, causing pulmonary edema and consequent deoxygenation of the blood, development of extensive edema in other parts of the body. What are the acute effects of a cardiac failure? If a heart suddenly becomes severely damaged, such as by a severe myocardial infarction, the pumping ability of the heart is immediately depressed. As a result, there are two consequences. The cardiac output would be reduced, and there will be damping of blood in the veins, resulting in increased venous pressure. What are the different compensatory mechanisms? As we know that the immediate compensatory mechanisms to compensate for the decrease in blood pressure, they are similar to the compensatory mechanisms which come into play when the heart is in a failing or a decompensated state. That is the baroreceptor reflex, the chemoreceptor reflex, and the central nervous ischemic response. The sympathetic system, it strongly becomes stimulated within few seconds, and the parasympathetic nervous system of the heart becomes inhibited at the same time. So after a few minutes of an acute heart attack, a prolonged semi-chronic state begins, characterized mainly by the two events, that is retention of fluid by the kidney and varying degree of recovery of the heart over a period of weeks to months. So what is the role of mitochondria? Why we are discussing mitochondria over here? As all of us know that mitochondria are a source of cellular energy. However, the function is critical for the heart because the heart needs an enormous amount of energy that is provided by the oxidative phosphorylation. Mitochondria create extensive network that it controls the metabolism. It is providing us with the ATPs. It is also essential for the removal of the eliminating the damaged organelles, and also for the production of reactive oxygen species, ROS. Any condition that leads to mitochondrial dysfunction will result in development of various cardiac illnesses, including the ischemic repercussion damage, atherosclerosis, hypertension, cardiac hypertrophy, heart failure, and diabetes. Also, other conditions are also related that can result in mitochondrial dysfunction, and that can result in producing different diseases. So coming to the importance of mitochondria, my slide is not changing, so I will go to my notes, that the mitochondria, they stop functioning, and there is some kind of dysfunctioning of the mitochondria. So can you play the slides or something? Yeah, I can't. Okay, first. Sorry. Any etiology of heart failure is linked with a gradual but continuous reduction in the activity of mitochondrial respiratory pathways, resulting in lower ATP generation capabilities. Energy deficit has been implicated as both a cause and consequence of heart failure. So if mitochondria, they are deficient, they are not working properly, or there is a mitochondrial dysfunction or disorder, the condition is known as primary mitochondrial disorder, PMDs, and they are a diverse category of diseases defined by altered mitochondrial function or structure as a result of mitochondrial or nuclear DNA abnormalities. The metabolic route most often impacted by the PMDs is the mitochondrial electron transport chain, ETC, that is the primary mechanism for generating energy like ATPs. So what happens is that the electron transport chain is disrupted, and that results in the dysfunctioning of the organs, and that results in oxidative stress and further dysfunctioning and cellular damage results. So these are the outcomes, that whenever there is disruption in the electron transport chain, there is decreased ATP, increased level of lactate, the CNS is affected, like the peripheral nerves, ears, skeletal muscles, kidney, heart, and endocrine organs. So symptoms could be seizures, encephalopathy, stroke-like events, ophthalmoplegia, hearing impairments, dementia, myopathies, and this includes metabolic function connection is significant in illnesses that can result in cardiac hypertrophy and eventually failure. So there are certain naturally occurring metabolites or naturally occurring things that we use in a dietary supplement that can help us in the restoration of the mitochondrial functions. And number one is the alpha lipoic acid. That is a natural antioxidant which is believed to have a beneficial effect on the oxidative stress reactions. It acts as a cofactor in the enzymatic reactions, and it will help in the generation of ATP. These are found in liver, meat, vegetable, fruits, and accessible as a nutrient supplement. Then we have arginine. It's also an amino acid. It helps as a nitric oxide precursor. It is which is used in vasodilatation and neurotransmitter. It also helps in the generation of ATP, and these natural supplements can help in patients in their syndrome, and they can be used in cardiac failure. Then we have got the carnitines. The carnitines is required for energy metabolism because it transports long-chain fatty acids in the mitochondria for oxidation. It aids in the amino acid and pyruvate oxidation and aid in the elimination of harmful acetyl coenzyme A metabolites by binding and excreting them in urine. Then we have got citrulline. Citrulline is found in watermelon. It's a very, very important substance. It is very, very helpful. It is also an nitric oxide precursor. It can help in the neurotransmitter formation and also the blood vessel formation. So the naturally occurring substances in our food, like Q enzyme, they can help in the participation of ATP generation and help in the processing of the transferred electron transport chain and protecting the mitochondria, preventing lipids, peroxidation, and DNA and oxidative damages. They are present in meat, whole grain, fish, bedside nutritional supplements. Then we have got feline acids, folates, folic acids. Jitne bhi hum khana khate hain, like green vegetables, they are rich in these phelinic acids, and they are also helpful in the generation of ATP, helping the mitochondria to regain its function. And then we have got niacin. It's also precursor of the coenzyme nicotinamide adenine dinucleotide phosphate, NADP, and NAD that are involved in a wide variety of biological redox processes.
Time is over.
Yes, I'm just finishing up. Then we have got riboflavin and thiamine, vitamin C, vitamin E, vitamin K. These are all helping the mitochondria cellular organelles to generate energy via oxidative phosphorylation. Thank you all. Thank you for listening, and let me present my presentation. Thank you so much.
Thank you so much. Now the next presentation is by Dr. Yusra Hamid, our postgraduate trainee of Al-Nafees Medical College. Topic is pathophysiology of myocardial infarction.
Everyone. I am Dr. Yusra Hamid, and our post. Topic of my discussion is pathophysiology of myocardial infarction.
Can all of you hear her?
Yes, we can hear.
Yes, sir, we can hear, sir.
Please continue.
Let's see what is myocardial infarction. It's basically necrosis of myocardium caused by insufficient blood flow either due to bolus, thrombus, or vascular spasm. It is commonly known as heart attack. Myocardial infarction is mainly due to underlying coronary artery disease. When any coronary artery is occluded, the myocardium is deprived of oxygen. Prolonged deprivation of oxygen supply to the myocardium can lead to myocardial cell death, eventually necrosis. Burden of this disease is very high as it belongs to the category of diseases causing maximum deaths worldwide. Risk factors of this disease include genetic predisposition, male gender, progressive age, smoking, obesity, high blood pressure, and high cholesterol levels, and sedentary lifestyle. Patients with this symptom sudden sharp chest pain, which is crushing in nature and being pain usually radiates towards the left shoulder, left arm, neck, and dorsal. Other symptoms include dyspnea, nausea, vomiting, dizziness, extreme weakness, diaphoresis, and sometimes denial. Coming to pathophysiology, but before moving to pathophysiology, I would like to summarize our knowledge about the blood supply of a heart, which is actually composed of left artery, left anterior descending artery, left marginal artery, right marginal, and right coronary artery. Left anterior descending artery occlusion is the most prevalent and documented. Now let's see how the occlusion occurs and how the obstruction of blood vessels occurs. On the left side of the screen, we can see normal human artery with normal physiology and all linings are intact. But if we move to the right side, we see at the center there is a lipid interspore with the outgrowth of abnormal outgrowth actually of the smooth muscle, topped with a layer of connective tissue which is enriched in collagen, which is called fibrous sac. Eventually, this whole thing leads to the formation of a plaque called arteriosklerosis. This plaque eventually enlarged and protrude into the vessel lumen can lead to acute coronary effusion, either by the formation of thrombus, embolus, or vascular spasm. Embolus is actually a dislodged portion of a thrombus which detaches from its primary site and starts to enter in the bloodstream and obstruct different smaller coronary vessels. With the help of a video, I would like to share this video for better understanding of pathophysiology of myocardial infarction.
Here you can see one of your arteries in cross-section with an atherosclerosis plaque which has developed over the years. Thrombosis occurs following a sudden rupture of this plaque. A clot of blood has blocked the coronary artery. It prevents blood and oxygen from reaching the part of the heart irrigated by this artery. This part of the heart, already injured, risks being completely destroyed without immediate intervention. Here you can see one of your arteries in cross-section.
Okay, to summarize the whole pathophysiology, I will take the help of this flow chart. Either because of arteriosclerosis due to arteriospasm or rupture of clot. All these conditions eventually lead to obstruction or occlusion of a blood vessel, leads to ischemia and hypoxia. This will speak experienced by the patient in a form of sharp pain, which is also called angina. If this ischemia is not treated and it prolonged for a long time, it leads to the formation of permanent thrombus, which causes necrosis and eventually myocardial infarction, which is the death of the myocardium. In this picture, we can actually see the progression of this. If artery is partially occluded, it will lead to less severe form of ischemia, and the 1/3 of the layer of the myocardium is detected. It's the formation of non-transmural ischemia or subendocardial ischemia, which depicts in ECG in the form of non-stent, or also called ST depression. But if the artery is completely occluded, or very less amount of the blood reaching to that area so that the myocardium is not able to maintain its function, all the 3 layers will be damaged, and this type of ischemia and infarction leads to the formation of transmural ischemia. This is depicted in ECG in a form of ST elevation. So for the diagnosis of myocardial infarction, the WHO has designed an early diagnosis criteria, which actually requires 2 of the following: typical clinical manifestations, ECG failures, and elevated cardiac biomarkers and cardiac enzymes. Moving to investigations, there comes the ECG, which is the mainstay and baseline investigation of the myocardial infarction. This actually shows the progression and evolution of the acute onset of myocardial infarction. Here we can see the normal physiological electrocardiogram picture, in which there is the R wave and upward T wave is present. There is no Q wave, but just after the onset of the disease, ST elevation is observed. Hours after the onset, we can observe the regression in the R wave. Q wave starting to appear. Few days after the onset, T wave started to invert and Q wave reversed. Some further days later, ST segment starts to normalize and T wave remain inverted. But few weeks after the onset of disease, ST segment and T wave becomes normal again, but Q wave worsens, which tells us about the chronic myocardial infarction happens to that patient. This is a graph which basically shows the cardiac biomarkers in myocardial infarction. Few important are myoglobin. Myoglobin is a partially elevated biomarker. Then troponin. Troponin is basically the most internationally recognized biomarker. Basically, it is more cardiac specific and sensitive. It starts to elevate 2 to 3 hours after the onset of disease. It reaches the maximum level at 24 hours after the onset of disease. Its level started to deplete 4 to 6 days after disease onset. This is a drop scale, which is used in our emergency department. Other investigation includes echocardiography, angiography, cardiac MRI, and chest X-ray. Moving to the management of the heart attack. So I will discuss just the basic or physiological displays, not going into detail of the treatment. After receiving a patient having the signs of ischemia and infarction, we give the first aid treatment to the patient and notify the respective hospital and emergency department to prepare for CPR and defibrillator. While doing so, we give the first aid treatment to the patient, which includes oxygen if saturation of the patient is low. Aspirin is given because of its antiplatelet function. Nitroglycerin is given to the patient either as sublingually or in the form of spray. Because it is a vasodilator and it helps to relieve the pains. Morphine is given to the patient, ease the patient to decrease discomfort. While treating the patient, simultaneously we try to obtain equal ECG elevation, because ECG will eventually decide the rate of the treatment. If we find ST elevation on the treatment, then we'll move to the reperfusion therapy without any delay. But if there is no ST elevation, then we will stick to atrial therapy along with invasive strategy accordingly if required. So, take-home message of the myocardial infarction or to survive a heart attack: minimize activity, seek medical attention as soon as possible, take aspirin and nitroglycerin accordingly and if appropriate. So, I want to summarize my whole presentation with a few recommendations and proposals that as a postgraduate student, I would like to it will introduce integrated options for undergrad students with the collaboration of all basic sciences departments that will be helpful for the student how to work in multidisciplinary approach, and introduce different workshops including APLS at undergrad level to enlighten the student with advanced knowledge for early diagnosis of the most lethal disease but without curable outcomes. Here are the differences. I thank you all for your patience.
Okay, well done, Dr. Yusra. Now the next, well, excellent. Now the last presentation is on pathophysiology of asthma by postgraduate trainee Dr. Aisha Mushtaq from Islamic International Medical College, Rifa University.
Assalamu alaikum. Am I audible?
Yes.
Yes, please.
Okay. And my presentation is on screen share as well?
No. इसको तो खत्म करो ना पहले यार हमारी हैं इसे खत्म करके दिखाओ ना कैसे ये नहीं होगी खत्म
इनको ऑफ करेंगे
So now is it visible?
यार इलियास इसे आप खत्म कर दीजिए ना ये थैंक यू जो नजर आ रहा है
डेस्कटॉप से भी खत्म करेंगे
आप अपनी शेयरिंग खत्म कर देंगे ना तो फिर वो नजर आएगी हाँ अब आप हमारी वीडियो दिखाएं बस हाँ आयशा डॉक्टर आयशा नाउ यू ट्राई दिस
So मैं ट्राई करती हूँ
स्लाइड ने दे दिया ऑप्शन पोज कर दिया
Sir, I think I don't have the access to share. So can you unshare the screen first? So let me open it first.
I think it is, we have given you the right to, you are the co-host right now.
So, I'm trying, sir. I think it will. Is it visible now?
No. No.
Not yet?
Not yet.
So it's coming on screen share on my side, but I don't know why it's not. So wait a minute.
Button, share screen, and then only.
So yeah, yes, I have done it, sir. So I have done it.
Because I did the same mistake, and then they told me that there is a button that you have to press, only then it will be shared. Because
Okay,
I thought I have shared it. Just try that.
So the one with the green that says share screen?
Yes.
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 Okay. Now it's visible?
No, no.
Yeah,
Yes.
it is available now.
No.
Yes, it is
Yes,
visible.
Visible.
Okay.
It is
Well
visible.
done.
Finally, sir.
Excellent, Aisha.
Sir, a new thing learned.
Yeah, this is what happened with me, I know.
So thank you for helping me out. So should I start?
Yep.
Yes.
So, Bismillahirrahmanirrahim. I'm Dr. Aisha Mushtaq. I have recently done my postgraduate training in physiology from Islamic International Medical College. And the topic of my today's presentation is the pathophysiology of asthma. So before starting up with my original thing, I would like to initiate with the norms. That is, all of you guys know that the process of getting oxygen into the body and releasing the carbon dioxide is called the normal respiration. So the normal respiratory rate in adults is 12 to 16 per minute. In the childhood, it is 15 to 25 per minute. In the early childhood, it is 20 to 40 per minute. And in the newborn, it is more, that is 30 to 60 per minute. So basically, the functional anatomy of the respiratory tract consists of the conducting passages. That includes the upper respiratory tract, including the nasal cavity, pharynx, and larynx, and the lower respiratory tract, that is the trachea, bronchi, and the lungs. So particularly, all of the pathophysiological diseases, basically, like the obstructive and the restrictive lung diseases, they basically involves the part of the lower respiratory tract, including the trachea, bronchi, and the bronchioles. So the airflow through the respiratory system is divided into three interconnected regions. One is the upper airway, the conducting airway, and the third one is the alveolar airway. So this is all you know, that the air travels to the trachea, then comes to the bronchi, then bronchioles, and finally the air enters into the alveoli, where the gaseous exchange takes place between the alveolar wall and the capillary wall. So the alveoli are the basic structural and the functional units of the lungs. So the structure of the respiratory unit consists of the respiratory bronchioles, the alveolar ducts, alveolar sacs, the antrum, and the alveoli. So this is the mechanics of the normal breathing. So once the person is preparing for the inspiration, what happens in the human body is the external intercostal muscles, they relax and the diaphragm relax. But during the inspiration, there is contraction of diaphragm, there is lowering of diaphragm, there is contraction of the external intercostal muscles, and there is elevation of the ribs. And during the expiration, there is contraction of the internal intercostal muscles, there is contraction of the abdominal muscles, and there is a return of the diaphragm, and there is the ribs and the sternums to the resting position. So basically, now the topic is regarding the pathophysiology of asthma that comes under the heading of the lung diseases. So basically, there are two types of lung diseases. One is the obstructive lung diseases, and the other ones are the restrictive lung disease. So the obstructive disorder is the reduction in the airway. So the shortness of breath occurs during the exhalation of the air, and the air will remain inside the lungs after the full expiration. As you can see in the diagram as well, that during the obstructive disorders, the forced expiratory volume in 1 second decreases. The forced expiratory volume, the forced vital capacity, it decreases. The ratio of the FEV1 to FVC decreases, but the total lung capacity and the residual volume increases. And the diseases that comes under the obstructive disorders includes the chronic obstructive pulmonary disease, asthma, and bronchiectasis. Now, the restrictive disorders, they are characterized by a reduction in the lung volume. So there is difficulty in taking the air inside the lungs. And as you can see in the diagram, that during the restrictive disorders, there is forced expiratory volume in 1 second increases, the forced vital capacity decreases, the FEV1 to FVC ratio also increases, but the total lung capacity and the residual volume decreases. And there is stiffness inside the lung tissues or the chest wall cavity. And the diseases included in this includes the interstitial lung disease, scoliosis, neuromuscular causes, and their marked obesity. So as you can see in this graph, that the compliance in the obstructive lung diseases, including the COPD and the asthma, it increases. And during the restrictive lung diseases, including the fibrosis and the pneumothorax, the compliance decreases. So the obstructive lung diseases, that includes asthma, chronic bronchiectasis, emphysema, and the airway obstruction. But my focus will be on the asthma. So asthma is the obstructive lung disease is basically the category of respiratory diseases that is characterized by difficult breathing with wheeze. There is airway obstruction. There is inflamed and easily collapsible airway, and it results from narrowing of smaller bronchi and large bronchioles. So the symptoms that the person experience includes the wheezing, tightness in chest, shortness of breath, chronic cough that may produce mucus, there is weight loss, there is gluten to the lips or the nail beds, and there is repeated respiratory infections. So basically, the obstructive lung diseases includes bronchial asthma, emphysema, and chronic bronchiectasis. So I would like to share a case presentation that how a person or a child can present in the OPD to the doctor. So there is a girl, Callie, a 10 years old white girl, and she comes up with the complaint and told the doctor that I feel it is difficult for me to breathe in deeply when I play with my friends. So the girl's mother started worrying about her breathing complaints 3 days ago. Kelly feels pain from time to time and cannot play with her friends all the time she wants. No medications were taken. So the past medical history suggests that there are no surgeries done with the child, no allergies, and pneumonia at the age of 4 does occur. And the social history reveals that she lives with her parents and the father is a smoker. So these are the types of the histories that we doctors come up in sitting in the OPDs when the patient comes up with any of the obstructive lung diseases. So asthma is basically a common lung-term chronic disease worldwide. It is the most common chronic disease in childhood, affecting an estimated 7 million children. And there are various triggering factors, including the inflammatory factors like the respiratory infections, the work you are in, and the allergens. There are irritants like temperature change, exercise, cold air, stress and emotions, and others includes the medications, any food additives that you use in your food, the pollutants you are exposed to, the gastric reflux disease if you have any, and the tobacco and the cigarette smoking. So the interplay between all these symptoms and the pathophysiology of the asthma starts that whenever you're triggered with any of these triggering factors that I have already told, what they do is that they causes the airways to get inflamed. And once the airway inflammation occurs, it leads to hypersecretion of mucus, airway muscle constriction, and swelling of the bronchial membranes. And all of these contributes to narrowing of your breathing passages, leading to the symptoms like wheezing, cough, shortness of breath, and tightness in chest. So as you can see clearly in this diagram that there is a normal airway that is more patent and compared with the bronchial asthma airways. So what happens in the bronchial asthma that it is caused by the contraction of smooth muscles and bronchioles, leading to obstruction of airways. And it causes obstruction is further exaggerated by edema of mucous membrane and accumulation of mucus and lumen of the bronchioles. So this is why the muscles, they get tight. There is less air flow through the trachea and the bronchi and the bronchioles. There is mucus infiltration. There is inflammation causing the narrowing airways. So this is what happens in the asthma. So during the attack, it is difficulty in the person to have breathing like inspiration and expiration. The bronchioles have tendency to dilate during inspiration, but they constrict during the expiration. There is more difficulty in experience during expiration.
Sorry, Dr. Aisha, time is over. Time is over.
Okay, ma'am, I'll just complete. Great effort is exerted by all expiratory muscles, causing compression of the chest, and it leads to the whistling sound, that is the wheeze. And the pathology and the pathogenesis of asthma includes that whenever the bodies or the cells are exposed to antigen, it produces the various immune responses, resulting in the production of various interleukins that mediates the inflammatory biomarkers like histamine, prostaglandins, leukotrienes, enzymes, leading to the bronchial hyperresponsiveness and airway obstruction, and the asthmatic symptoms includes. So the effect on the lung volume capacities, as I've already told you, that the functional residual capacity increases, the tidal volume, the vital capacity, and the forced expiratory volume in 1 second decreases. And the FEV1 is reduced due to an obstruction of air escaping from the lungs. And the ratio of FEV1 to FVC ratio is less than 0.7. That is roughly about 75%. So this is the FEV1 ratios in the obstructive restrictive lung diseases. So this is a gold standard that is where that can be predicted by the FEV1 percentage that can be predicted by the gold standards. And this is the chart showing it. So there are various types of asthmas like allergic, seasonal, occupational, non-allergic, exercise, difficult asthma, and brittle asthma. So these are the various severities and the conditions, including the intermittent asthma. That is, if you are suffering from less than 2 days a week, your lung functions are normal in this, the person can do the normal activities, and you can use the quick relief medicines like albuterol 2 or fewer days per week. So when you're suffering from the mild type of asthma, that is the symptoms that last for greater than 2 times a week and wake up 3 to 4 nights a month, a person may have affected normal activities, lung function is normal, and the quick relief medicines can, if you take it more days per week, 2 or more days per week, can do a great response. And in the moderate, it is you wake up at 1 to 2 times nights per week. Almost the symptoms are every day. The person's normal activities are limited, lung functions are decreased, and you can use quick relief medicines. Whereas in the severe form of the asthma, the symptoms during every day, wake up every night, person has extreme limitations on normal activities, there is significant decrease in lung functions, and you have to take the quick relief medicines almost every day. So the doctor can ask you questions like, how often do you have symptoms? How often do you wake up at night from coughing? How often do you have trouble breathing? Do you use the inhalers or the quick release medication? Do you have to go to the emergency, and how you manage your asthmatic symptoms? So the next protocol is diagnosis of the asthma on the various clinical lab tests, which includes the pulmonary function test, We can do the FEVC test, FEV1 test, and the ratio test. So these are the lab reports that you can assume on the obstructive, restrictive, and normal. So the other tests includes the fractional exhale nitric oxide test, bronchial provocation or trigger test, allergies test, and there is various blood tests that can be done for the allergens. The chest X-ray can be done, computed tomography scans, and bronchoscopy is the ultimate regime to diagnose the asthma. So asthma attacks, basically, there are sleep problems, daytime sleepiness, fatigue, and lack of exercise. Many people with asthma have GERD, frequent respiratory infections, complications from flu and pneumonia. And if left untreated, it can lead to serious health complications. And if someone has sleep problems and fatigue due to asthma, they may experience unwanted weight gain, leading to conditions like diabetes, heart diseases, high cholesterol, high blood pressure, and stroke. So this is all what I have already told. This was a recap and the flow chart. So coming towards the treatment, so the basic treatment regime for the asthmatic patient is the 40 to 60% of the oxygen, high doses of inhaled bronchodilators, systemic corticosteroids can be used, IV infusions can be given, and subsequent management. So the oxygen therapy is that 92% of the high concentration oxygen should be given as a assisted ventilation regime to the asthmatic patient. And the inhaled bronchodilators are the salbutamol 5 milligrams per hour can be used. You can use nebulizers driven by oxygenation, metered dose inhalers through a space device, and inhaled anticholinergics like ipratropium bromide 0.5 milligrams can be used. And regarding the systemic corticosteroids for hydrocortisone, sodium succinate in 200 milligrams 4 hourly intravenous can be done if the patient is unable to swallow it. And the arterial blood gases, we should correct ABG, especially if the patient has partial pressure of carbon dioxide more than 7 and partial pressure of oxygen less than 8, and the patient deteriorates. So if the patient fails to improve intravenous magnesium sulfate, B2 agonist and aminophylline can be used, and the chest X-ray can be repeated to exclude that the person has not suffered from the pneumothorax. So this is another treatment regime, and these are the types of the inhalers that are available in the market. So the lifestyle modification can be done to reduce the maximum deterioration that is done by asthma. That includes reducing exposure triggers, reducing your stress, eating healthy food, breathing deeply, doing the exercise, take a good sleep, reduce the stressful situations, change the negative thought, have the positive affirmations, and relaxation. And these are the various food modalities that can be used as a predisposition that you should not suffer from the asthma. These are the references. So I would like to end up my presentation with this beautiful quote from Joseph Pilates that says that breathing is the first act of life and the last. Our very life depends on it. Since we cannot live without breathing, it is tragically deplorable to contemplate the millions and millions who have never mastered the act of correct breathing. So thank you very much, and thank you for giving me an opportunity to present on this forum.
Thank you so much, Dr. Aisha. Wonderful presentation. Now the time is for question answer session. Okay, thank you so much, Dr. Aisha. Question answer session time. Finish? No. Sir?
Thank you very much, all the presenters and participants, for this wonderful seminar. At this point, I would like to announce the next PPS seminar. The topic will be pathophysiology of diseases related to gastrointestinal, hepatobiliary, renal, and body fluids. That will be, inshallah, on 16th August 2023, Wednesday, from 10 a.m. to 12 noon. Every physiologist is most welcome to participate, yourself as well as your students. They all are welcome to participate in this forthcoming seminar. The sheets for today's seminar will be sent to all participants on their addresses. And that's all for today's seminar. Thank you, everyone, again.
जज़ाकल्लाह खैर जी, बहुत बहुत शुक्रिया सब लोगों का. Thank you very much.
Thank you very much, sir. And
Thank you, sir.
to all
Thank you,
the
sir.
respected,
Jazakallah.
Thank you, sir. Thank you very much.
all the respected members, both
Sir, the President, Sir Umar Ali Khan, now distribute the sheet to the participant. Dr. Yusra. Yes, sir.